Dear student friends, I am going to take a class regarding the proteins. I am telling the fundamentals of proteins, what are the basic requirements for proteins, the structure elucidation, classification of proteins, and color test. This age is called the polymer age. Proteins are biopolymers. Polymers are divided into two, non-biopolymers and biopolymers. Proteins are biopolymers. Proteins means proteus. The word, the Greek word means first. In the world, every organisms consists of the carbon and hydrogen compounds. Here the proteins are one of the compounds like that. Proteins are biopolymers. Definition for proteins. Proteins are complex nitrogenous compounds which are found in the protoplasm of all animal and plant cells. Proteins are biopolymers, contains large number of amino acids joined to each other by peptide bonds. It is also called as the condensed biopolymers, consist of peptides and polypeptides. The biological importance of proteins. The biological importance consists of the protein comprises the structural building blocks as well as the most of the functional machinery of living organisms. Protein constitute the structural units of protoplasm. Certain proteins are responsible for the control of expressions of hereditary information from its first transcription from the gene to its final translation into polypeptide chains. Proteins are primary source of amino acids. The biological catalysts called enzymes are made up of proteins. The proteins are responsible for the functions of respiration, muscular contraction, active transport of cellular constituents, electrical transmission, etc. Proteins are hormonal regulators of metabolism and its storage depots of certain molecules. The antibodies are complex proteins. These serve as a weapon in the reference arsenal of the organism. The example is virus. Nucleic acids serve as the enclosure which is occupied by the nucleic acid in the infective agent. Life in the supercooled water. Proteins that has a remarkable capacity to lower the freezing point of the fish's blood. Buffer activity. The buffer activity control of pH in biological system is done by the proteins. Molecular weight. Proteins are macromolecules which are constructed by the repetition of one or more structural elements called the monomers. In the protein, more than 20 amino acids present. The monomers contains more than 20 amino acids are called proteins. These proteins are classified as the small molecules called the polypeptides, peptides and amino acids, the final component. Every protein contains the final product is the amino acid. The peptides and polypeptides are condensation polymers of polyamides. It contains the polyamide groups. Classification of proteins. In the simple amino acid, the glycine contains these NH2 CH COH R is the basic unit of the acid. Proteins are classified according to the structure, solubility, behavior and non-protein moiety present in it. According to the solubility, it is divided into two, that is called fibrous protein and the globular proteins. Those proteins soluble only in concentrated acids and alkalis are called as fibrous proteins because they are contains the fibrous materials. The examples are silk, wool, skin, hair, horn, nails, quills of connective tissue, bones, etc. The list is going on. The second one is the globular proteins which are soluble in dilute acids as well as bases. The enzymes, oxygen carrying proteins, hormones, etc. are coming under this group, globular proteins. The next classification of proteins is structural proteins. Simple proteins are hydrolyzed to polypeptides, peptides and finally the amino acids. The simple proteins are albumin, globulin, Glutenin is the wheat enzyme and the arginine is the rice enzyme. Albuminized scleroproteins are keratin, hair, hoof, silk, etc. In the simple proteins, these are further classified into the neutral proteins, example alanine. The basic proteins, example is arginine. The acidic amino acids are called the aspartic acid, the amides that is called aspargine. Examples for the simple proteins, they are comes under various organisms present in the human body. The conjugated proteins, the non-protein substance called the prosthetic group is present. Here there are a number of prosthetic groups are present. One is the nucleoproteins. Here the nucleic acid is present. The chromoproteins, the colored prosthetic group is hemoglobin. Glycoprotein, glycogen is a prosthetic group. Carbohydrate, mucoprotein or egg albumin, serum albumin are examples for glycoproteins. Phosphoproteins, phosphoric acid is an example for the phosphoproteins. Lipoprotein, serum is the example for the lipoprotein. Metalloproteins, here we are giving two types of metalloproteins. One is the hemoglobin, the other one is myoglobin. Then we have to go for the some of the physical properties of proteins. The first one is the denaturation. Suppose the proteins are irradiated with optical activity and precipitated from aqueous solution by heating. The proteins are standing in acid or alkali or treated with ethanol or acetone or high concentrated urea detergents. The proteins in these solvents may be precipitated in state are said to be denatured, and the process of reaching this state is called the denaturation. Occurs nearly in the isoelectric point. Denaturation is now considered to be the result of changes in conformations or unfolding of the protein molecule, that is the secondary and the tertiary structure is changed. The renaturation, the opposite of the denaturation, called annealing. The irreversible or uncontrolled denaturation is the boiling of egg. Secondary and the structure is changed or destroyed. Then the isoelectric point. Normally the proteins are neutral in nature in our human body. Here, but in proteins there are two kinds of ionizable groups are present. One is the NH2 and another one is COOH group. Both are easily ionizable. Then how it is possible to be neutral in nature, the proteins? Here we have to come across that the proteins may be present in the form of the Zwitter ion, that is NH3 plus or CHCOO minus or NH2 or CHCOO minus. So it may form the inner salt or the Zwitter ion or dipolar ion in a particular stage, that is called the isoelectric point. In the isoelectric points there is minimum, the physical properties are minimum, mobility of ion in the electric field is slow, the osmotic pressure, swelling capacity and solubility are minimum, coagulation are also minimum in nature. Then the color reactions. Proteins are detected or determined using various color reactions. There are different types of color reactions. One is the biuret test. In the biuret test, the protein is treated with alkaline solution and warmed with dilute copper sulfate, so that we get a red or violet color. This reaction indicates the presence of the group, the polyamide group CONH or CH or CONH. This polyamide group, presence of the polyamide group is confirmed through this biuret test, but this test is not useful for the dipeptides. The next one is xanthoprotic test. Proteins are treated with concentrated nitric acid and warmed, gives yellow precipitate. When it is yellow color is treated with alkaline, we get orange precipitate. This test is due to the nitration of benzene ring in phenylalanine, tyrosine and tryptophan. Suppose some nitric acid are fallen in our skin, it may become yellow. This is because of this reaction, this xanthoprotic reaction. Then the third one is the Millen test. Mercuric nitrate in nitric acid containing a trace of nitrous acid is treated with proteins, gives a white precipitate, which is on heating gives a red, indicates the presence of proteins. Test is a characteristic test for the phenolic proteins. Here the phenolic protein, the only phenolic protein available is tyrosine. Then the Nin-Hedrin reaction. Here the Nin-Hedrin 1,2,3 thione is used to test for the different functional group that is present in the proteins. Using this test, we have to conclude a blue dye is obtained in the final reaction, which gives the presence of the NH2 or N group nitrogen in the protein is confirmed. In the proteins, the end group analysis is very important one, because in the end group, which group is attached to the particular protein may be determined, because in the end group, whether it ends with the COH group or the NH2 or some other substituent like phenol or any other thing may be determined by this test or this analysis. Here the C-terminal amino acids, there are two types of end group analysis. The first one is C-terminal amino acid determination, and the next one is N-terminal amino acid determination. The first one is C-terminal amino acid determination. Here the test is called hydrogenolysis. Hydrazine is used for this test. The end product is hydrazine compound. Using column chromatography, strong cation exchange resin on elution, the strongly basic hydroxides are retained, but the free amino acid is eluted and can be identified which compound is present in that particular protein. Then using reducing agents like lithium aluminum hydride and lithium borohydride, we get the alpha amino alcohols, forming alpha amino alcohols, which on hydrolysis give the alpha acids, alpha amino alcohols. This can be separated using paper chromatography. Then carboxypeptidase, the free carboxyl group is obtained using this test. On hydrolysis, we get the successive terminal amino acids. The N-terminal amino acids, amino acid analysis. This method is called the Edman method. Here the phenyl isocyanide is used to determine the N-terminal amino acids. Here thiocyanate is treated with the amino acids, which will give phenyl thiocarbamyl, which on hydrolysis give the phenyl thiohydranidine, that is NTAA, the N-terminal amino acid, which can be determined by the paper chromatography method. Then the another method is the DNP method. Here the amino acids is treated with 1,fluoro,2,4-dinitrobenzene in the presence of sodium bicarbonate at room temperature, gives the, which are treated with the hydrogen fluoride, which are treated with an acid which gives the alpha amino acids and the 2,4-dinitrophenyl amino acid. Today's world is a polymer era or plastic age. Here the biopolymers, protein is very important in nowadays. Even today the polymer protein chemistry is enormously growing. This process is enormous as well as a great one. Biopolymers makes the world a very great one. Here in this class I am given only a brief introduction about the fundamentals as well as the structural elucidation, peptides, polypeptides, and the color reactions of proteins, classification of proteins, and some physical properties like the isoelectric point, denaturation, etc. These things are only fundamental in nature. We have to refer enormous amounts of materials for this to obtain very good result.